Hi everyone, and welcome to the lecture on the autonomic nervous system. The nervous system is divided into two parts: the central nervous system, or CNS, which is comprised of the brain and spinal cord, and the peripheral nervous system, or PNS, which is comprised of the somatic and autonomic nervous system. The somatic nervous or conscious nervous system consists of peripheral nerve fibers that send sensory information to the central nervous system, and the motor nerve fibers that project to the skeletal muscles. Today we focus on the autonomic or unconscious/involuntary branch of the nervous system. The autonomic nervous system is divided into three parts: the sympathetic, parasympathetic, and enteric nervous system. Collectively or altogether, the autonomic nervous system controls smooth muscle of the viscera, aka internal organs, as well as all of our glands. As we learned in the previous week's PowerPoint on the peripheral nervous system, there are incoming afferent sensory neurons from the peripheral nervous system that move into the central nervous system. And then we have neurons that are carrying impulses away or out of the central nervous system, the motor or efferent neurons. Unlike somatic motor neurons, which conduct impulses along a single axon from the spinal cord to the neuromuscular junction, autonomic motor control involves two neurons in every pathway. So, general organization of the autonomic nervous system is as such: the preganglionic neuron, the one that's going to carry information into the back of the spinal cord, is located in either the brain or the spinal cord within the gray matter. The preganglionic neuron projects to an autonomic ganglion. Remember, a ganglion is a collection of neuronal cell bodies outside the central nervous system. And then coming out of that, the postganglionic neuron, the one that was synapsed onto, then projects to the target organ. Smooth muscle is the major target of the autonomic nervous system. Smooth muscle is located in all internal contractile organs except for the heart, which contains a specific type of muscle known as cardiac muscle. Under the microscope, smooth muscle has the following features: very small spindle-shaped cells, no striations or stripes. They do contain actin and myosin, but it contracts a little differently, kind of like a pair of the Chinese handcuffs that some of you may remember getting in birthday bags, as opposed to skeletal muscle. And smooth muscle is less organized than skeletal muscle, meaning it's not organized into those sarcomeres, which create the visible bands. Smooth muscle, in order to contract, does have to contain actin and myosin. However, even though the filaments are essentially the same in smooth muscle as they are in skeletal and cardiac, the way in which they are arranged is different. Some of the regulatory proteins also differ, and there are some specific smooth muscle isoforms of both actin and myosin. Smooth muscle does not contain the protein troponin. Instead, the molecule calmodulin takes on this regulatory role in smooth muscle. Actin filaments attach to other actin filaments via structures known as dense bodies, which act much like the Z-lines or Z-discs in striated skeletal muscle. In smooth muscle contraction, calcium ions enter the cells from the extracellular fluid as well as from intracellular stores. This leads to a series of biochemical reactions taking place, which leads to the myosin cross bridges being phosphorylated and in turn binding to actin. There's single unit smooth muscle versus multi-unit. Multi-unit smooth muscle organs are ones in which each smooth muscle cell acts relatively independently of the other smooth muscle cells within that organ. In this sense, multi-unit smooth muscle is like skeletal muscle. Multi-unit smooth muscle is found in large blood vessels, large airways of the lungs, the ciliary muscle of the eye, the iris of the eye, and the erector pillae muscle, which gives us goosebumps. Multi-unit smooth muscle tends to have fewer gap junctions between cells, making each cell operated independently. Multi-unit smooth muscle also tends to have higher innervation by the autonomic nervous system, which in greater ratios than in visceral smooth muscle. Single unit smooth muscle organs are also called unitary or visceral smooth muscle. Single unit smooth muscle is found in the hollow organs of the digestive, reproductive, urinary systems, as well as in the smaller blood vessels. In contrast to the cells of the multi-unit smooth muscle, which behave somewhat independently of one another, the cells in single unit smooth muscle behave in a syncytial manner, much like cardiac muscle. So gap junctions, the literal physical connections between adjacent muscle cells, are much more abundant. And as a result, the cells contract as a single coordinated unit. So because of this, innervation tends to be relatively sparse, not needing all that independent nervous innervation. Single unit smooth muscle is known as myogenic, which means that certain muscle cells are self-excitable, which means that they do not require nervous stimulation in order to contract. Instead, they experience automatic, spontaneous shifts in ion concentrations between the extracellular fluid and the intracellular fluid, which cause spontaneous, regular depolarizations to threshold. And these can come in two forms: pacemaker potentials seen in the heart, as well as slow wave potentials. The rate and strength of contraction of single unit smooth muscle is influenced by the autonomic nervous system, hormones, as well as a variety of other factors. All of these influences alter the level of the calcium ions in the cell's cytosol, making more or less frequent those spontaneous depolarizations and resulting contractions. Now, unlike skeletal muscle, where there's no gradation of contraction, smooth muscle can have gradations of contraction, and this depends on the level of calcium in the cytosol of the muscle cells. Many cells have enough calcium in the cytosol to maintain tone, or a low level of tension, in the absence of action potentials. Smooth muscle can also exist at many different lengths without a change in tension. Connective tissue on the outside usually limits the stretch, which is important for the emptying of hollow organs such as the bladder. Contraction is also very slow and energy efficient since it needs to happen pretty much constantly. Now, as far as the autonomic nervous system, we know that it innervates cardiac or heart muscle, smooth muscle, and both exocrine and endocrine glands. The two autonomic nervous system neurotransmitters are acetylcholine and norepinephrine, aka noradrenaline. Each autonomic nerve pathway is comprised of two neurons, a preganglionic neuron and a postganglionic neuron. Now, we will not focus on the enteric nervous system, but rather going forward in this lecture focus on the sympathetic and parasympathetic nervous systems. The sympathetic nerve fibers originate from the thoracic and lumbar levels of the spinal cord, so basically the chest and lower back regions, whereas the parasympathetic nerve fibers originate up high from the cranial region in the skull, as well as the lowest level, the sacrum of the spinal cord. The parasympathetic division of the of the autonomic nervous system controls what are known as the rest and digest responses, or feed and breed responses. The parasympathetic nervous system works to conserve energy. It lowers your blood pressure and slows your heart rate, and allows for steady, slow digestion to start and occur. The cell bodies of the parasympathetic neurons are located in the spinal cord, the sacral region, low, and then up in the medulla of the brainstem. In the medulla, the cranial nerves 3, 7, 9, and 10 form the preganglionic parasympathetic fibers. The preganglionic fiber from the medulla or spinal cord projects to a ganglia very close or on the target organ and makes its synapse. This synapse uses the neurotransmitter acetylcholine. From the ganglion, the postganglionic axon projects to the target organ and uses acetylcholine again at its terminal. So in the parasympathetic division, both the preganglionic and postganglionic neurons use acetylcholine as the neurotransmitter. The sympathetic division of the autonomic nervous system controls the fight or flight responses. Instead of conserving energy, it uses energy. Blood pressure increases, heart beats faster, and digestion slows down or stops. The sympathetic nervous system originates in the spinal cord. Specifically, the cell bodies of the first, or preganglionic neuron, are located in the thoracic and lumbar regions of the spinal cord. Their axons from these preganglionic neurons project to a chain of ganglia, or neuronal cell bodies, located right near the spinal cord. In most cases, this neuron, the preganglionic, makes a synapse with another neuron, the postganglionic, in this ganglion. The postganglionic neuron then projects to the target, either a muscle or a gland. The synapse in the sympathetic ganglion uses acetylcholine as the neurotransmitter from the preganglionic onto the postganglionic, and then from the postganglionic onto the effector, the target muscle or gland, it uses the neurotransmitter norepinephrine. The autonomic nervous system controls the involuntary or unconscious responses of the visceral organs. The visceral organs receive dual innervation, meaning that they have input from both the sympathetic and parasympathetic branches of the autonomic nervous system. This leads to functional antagonism, kind of like a brake and a gas pedal in a car. Usually, both subdivisions are active in controlling the activity of an internal organ, but depending on what is going on, either sympathetic or parasympathetic tone will control. There are a few exceptions to this. Most innervated blood vessels, primarily the arterioles and the veins, are innervated only by sympathetic fibers, because if they were innervated by parasympathetic, the tone in their walls would become so low that blood would cease to pump. Another exception are the sweat glands. These are innervated only by sympathetic nerves, so you can either sweat a little or a lot, but it's like you can't have negative sweating, if you will. And then finally, the adrenal medulla, the inner part of the adrenal glands, is only sympathetically innervated. Now, again, we're going to focus primarily on the parasympathetic and sympathetic branches, so you can kind of forego the adrenal medulla here. Each autonomic nerve pathway, as we've stated numerous times, has two neurons, a preganglionic and a postganglionic neuron or fiber. In the sympathetic nervous system, the preganglionic fibers are short. They synapse onto the cell body of the postganglionic neuron in that sympathetic chain ganglia, which lies right to the side of the spinal cord. By contrast, the postganglionic fibers are long, terminating on the effector muscles or glands. In the parasympathetic branch, the preganglionic fibers are long, traveling to the postganglionic neurons that sit either near or on the effectors. Ganglia, again, are groups of neuronal cell bodies located outside of the brain and spinal cord. Sympathetic ganglia are located right near the spinal cord in the sympathetic trunk or sympathetic chain ganglia. Parasympathetic ganglia are located near or on effector organs. Acetylcholine is released from cholinergic neurons or fibers and is a neurotransmitter. It's the only one used in the parasympathetic branch, and it's used by the preganglionic sympathetic fibers. Norepinephrine or noradrenaline is released by adrenergic fibers, and it is released by postganglionic sympathetic nerve fibers. Now, acetylcholine binds to two different types of cholinergic receptors. Nicotinic receptors are located postsynaptically in all autonomic ganglia, as well as at the neuromuscular junction. At these junctions, nicotinic receptors function as the excitatory receptor for the postsynaptic cell, meaning that when they bind to acetylcholine, they go on to send a nerve impulse. Release of a sufficient quantity of acetylcholine from the adjoining presynaptic cell causes an excitatory response in the autonomic ganglion cell bodies and in somatic muscle fibers of the neuromuscular junction. Muscarinic receptors for acetylcholine are located postsynaptically at the parasympathetic neuroeffector junction. At these junctions, muscarinic receptors function either to increase or decrease the activity of the postsynaptic cell. Adrenergic receptors bind to norepinephrine or noradrenaline, as well as epinephrine/adrenaline, released from sympathetic postganglionic neurofibers. Alpha-1 adrenergic receptors have an excitatory response in the effector organ, whereas alpha-2 receptors mediate an inhibitory response. The muscle does not contract or the gland does not secrete. Beta-1 receptors are found primarily in the heart and mediate an excitatory response, meaning that when they bind to norepinephrine and epinephrine, they cause the heart to beat faster. Whereas beta-2 receptors, when they bind to adrenaline, they reduce the contractility of the heart. They have an inhibitory response. The adrenal glands, I always say, are like an M&M with an outer candy shell that we know as the cortex, which releases a variety of different hormones, and then the inner medulla is a modified ganglion or cluster of neuronal cell bodies, which releases 80% epinephrine or adrenaline and 20% norepinephrine or noradrenaline. Sympathetic responses include the pupils of the eye dilating, sweating is promoted, heart rate increases, the bronchioles dilate, motility for digestion is inhibited, the liver converts the storage form, glycogen, into usable monomers of glucose, urination and defecation are inhibited, and we prepare for emergency or stressful situations, hence flight, fight, or fright. When you think about it, I want you to think of yourself as an animal instead of a human. Think about a zebra out on the plains of Africa running from a predator. The pupils of your eye dilate to allow you to see better. Sweating is promoted to cool you down as you run for your life. Your heart rate increases to increase blood and nutrient and oxygen delivery to your muscles and your heart. The bronchioles dilate, allowing you to bring in more oxygen, and we do not digest or urinate because we don't have the time or energy for that. We break down those storage forms. Remember, glucose is kind of piled up like Tupperware containers are into a stack known as glycogen. As mentioned in the previous slide, the sympathetic nervous system collectively works to increase one's cardiac output, the amount of blood pumped with every heartbeat, increase pulmonary ventilation, the bringing of oxygen and nutrients into the blood, and the exhalation of CO2 and waste products out, increasing blood flow to skeletal muscle, and an overall increase in available usable energy in the form of glucose. It also decreases the function of non-emergency systems such as digestion and reproduction. And important to note, the sympathetic nervous system responds as a whole, meaning that if your heart rate is going to increase, so is inhibition of digestion going to take place. It's not just parsing out certain responses, it's all or nothing. The parasympathetic nervous system responses are usually the opposite of that of the sympathetic, meaning decreased heart rate, or no response, meaning no activity of glucose production from the liver. The parasympathetic nervous system reigns supreme during quiet, relaxed times, hence the rest and digest or feed and breed notation. Parasympathetic effects include our normal housekeeping, meaning obtaining nutrients and energy by digesting food, promoting secretions and mobility of the digestive tract, eliminating wastes in the form of urination and defecation. The parasympathetic stimulation, unlike the sympathetic, occurs as needed and not all or nothing. So here's a nice summary of the various body structures and how they're affected by both sympathetic versus parasympathetic stimulation. And this does a really good job of pointing out how the parasympathetic nerve fibers arise from the cranial portions of the medulla, as well as the sacral regions of the spinal cord, and the various organs that they innervate and the responses that they mediate, whereas the sympathetic nerve fibers arise from the thoracic and lumbar regions. You see in the synapse in the sympathetic trunk, and then it shows the various organs and responses mediated by the sympathetic fibers. The two branches, the sympathetic and parasympathetic branches of the autonomic nervous system, definitely interact with one another in a dual innervation dynamic antagonistic way. This includes dynamic antagonism of heart rate, the respiratory rate, and the GI tract motility. Sympathetic tone reigns supreme in the vascular system, where we want there to always be a somewhat constricted level to the blood vessels, whereas parasympathetic tone dominates the heart, the gastrointestinal tract, and glands, except for the adrenal and sweat glands. In the somatic nervous system, the effectors are skeletal muscle. In the autonomic nervous system, the effectors are smooth muscle, cardiac muscle, and glands. The somatic nervous system only has a one neuron chain, as seen in the upper diagram here, whereas in the autonomic nervous system, there are always two neurons in the chain. The preganglionic is the primary, with the cell body located in the brainstem or spinal cord, whereas the ganglionic or postganglionic neuron is the postsynaptic or secondary, with the cell body in an outlying ganglia and the axon innervating the target organ or gland. Somatic nervous fibers are seen only in the central nervous system, whereas in the autonomic nervous system, we have the preganglionic fiber originating in the central nervous system, but the cell body of the postganglionic in a ganglia or collection of cell bodies outside of the brain and spinal cord. In the somatic nervous system, the only neurotransmitter used is acetylcholine, and it always causes excitation of the effector, whereas in the autonomic nervous system, the preganglionic and postganglionic of the parasympathetic release acetylcholine. In the preganglionic, in both the sympathetic and parasympathetic, acetylcholine always leads to excitation of the postsynaptic or postganglionic neuron. In the parasympathetic, acetylcholine can either be excitatory or inhibitory on the postganglion or the effector, whereas in the sympathetic branch, the postganglionic neuron always releases norepinephrine and it's always excitatory. So to conclude, this is a great diagram, right? It shows in the parasympathetic nervous system, the preganglionic fiber has to travel a long distance to synapse at a ganglia that's either on or near the effector organ. The first preganglionic neuron in the parasympathetic branch releases acetylcholine onto the cell body of the postganglionic neuron, which in turn also releases acetylcholine onto the effector. In the sympathetic branch, by contrast, the preganglionic fibers are much shorter and synapse with the cell body of the postganglionic neuron in a close-lying ganglia called the sympathetic trunk or sympathetic chain ganglia. The postganglionic fiber then releases norepinephrine onto the effector. And here again we have a summary slide showing the various outputs of the somatic versus autonomic, so the conscious versus the unconscious nervous system. Another diagram that we've seen before. And finally, we have the cervical plexus, which supplies the back of the head, the neck, and the shoulders. And we've talked about this in the previous diagram. The phrenic nerve is formed by fibers from C3 through C5, lies on the scalenus anterior muscle, and enters the thorax by passing between the subclavian artery and vein and pierces the diaphragm to supply it so it can contract and mediate breathing. If the phrenic nerve is severed, we will suffocate. So if someone breaks their neck, they can die by suffocation. And again, this is just a review of the previous diagram. The brachial plexus serves the arm, the forearm, and hand, and is fed by C5 through C8 and T1. The lumbar and sacral plexi merge to form the lumbosacral plexus, which serves the anterior branches of this plexus supply the flexor muscles of the limb. The posterior branches supply the extensor and abductor muscles.